when they're not there. I don't see them either.
So this one's making me scratch my head even more. So even more exciting that we can maybe take half of it.
Okay.
The goal is to get some branches that have branches coming off of them, so that if there are those nodes, they would be in the sample.
Right. Yeah, this is, looks like it might be difficult to get enough of the base without getting all of it.
Wrist drift.
Oh, it is. Yeah, it's smaller than I thought. So.
Can you zoom out for a second, Wayne?
Oh, sorry.
Just to
out.
make sure I'm not smacking the camera here.
You had it right before.
Did I?
Yeah, you want the cutters.
I do. Thank you.
Root side, and then the Tygon will grab your sample, bring it up.
Yeah.
I'm gonna have to play with this thing for a minute. Which one is it?
Okay.
I want the other way, right?
No, I think it was
You
the
were
other
right
way.
before. The cutters go root side.
That.
So the part we're going to leave have the cutters on it, and then the Tygon grabs the sample.
Okay.
Where are you going, Herc? Stay. Okay, you can zoom a bit there, Wayne.
All right.
And so how big a piece are you looking for here?
Small. I'm sorry, Herc, what was that?
How big a piece do you want?
How big a piece, Danny? I'd said half, or at least half.
At least half. Can you zoom out just a bit, Wayne?
Yep,
so I can
coming
see
wide.
That's good right there, man.
All right, staying there.
Yes.
So.
And then as close to the base as you can, so we get multiple branches.
Is that too much, or?
No.
I think that looks great.
Okay. So cut. Let me try something here. I don't want to get the.
Ooh.
Oh, the whole thing came off.
Yeah, I think you got it all.
Yeah, you got the whole thing.
Is that too much?
No, that's okay.
Sorry.
It donated itself to us.
Is it still the same?
Okay, hold on. Zoom in. And let's give it some light and get a good focus. Okay, that's it.
Last time I did that, it was a 1,000-year-old black coral the size of a Christmas tree.
And you took the whole thing.
Oh, complete pandemonium.
Oh, no.
Do you want any rotate or any other close-ups here?
We can get some of this. So Danny's back, and we can get some shots of it in the minute.
Okay.
But the polyps are in now, so it's not as helpful, but we can see the branching pattern.
Yeah, there's
there.
definitely no black bands.
Yeah.
No. And
what bio box do you want this guy in?
Oh, it's not branching though.
that's a good shot. Okay, coming wide.
You're going to have to come off the wall if you put it in the front porch.
What's that?
All right, let's...
Why?
I said, what's that?
I said you'll probably have to come off the wall if you're going to put it in the front bio box.
you want to...
Science, where is this?
Yeah.
Where do you want your sample stored?
So we have the forward bio boxes. A and B are both empty, or if you wanted a standard bio box, you can put it in B or C to keep it separate from the other corals.
I can't hear you back there, guys.
Can you hear me?
Hold on. Sorry, who is talking?
This is the data logger. Can you hear me?
Let me give you a little more.
Okay.
Okay, try talking now.
Yeah, so you have the forward bio boxes, A and B both available. There's no specimens in there.
Okay.
You could put them in there, in one of those. Or you also have the standard smaller boxes, B through D.
Go for the forward one since I'm up against the cliff here. Do you want to bump that sample tray out?
Sample tray coming out.
Tool tray.
Tool tray,
It's
I
tool
mean,
tray,
sorry.
right,
Full wide.
Roger.
Lambda and omega, huh? You did that.
Could do a touch more. When it hits the cliff, stop.
Trying
You can
not
see
to.
it bubble there.
Yeah, I see it.
Okay, good. Your choices are lambda or omega. So that dealer's choice here, is it?
Set 2 and hurt lobe. Alright, and set 3. Okay.
Guess so.
Lambda it is.
Lambda.
Yeah, it's kind of low-hanging fruit for me at this angle.
Ooh.
Might be in the bumper bar there.
Okay.
Go for a track, see what happens.
Okay.
Okay, hold it there.
It's pushed.
The
Can you
Zeus
come out a
rack
little?
back.
It is.
Want me to come out a little?
Yes,
little?
please.
Okay, coming out. Clear.
Okay, retract this to here.
Okay. Moving.
Oh, come on.
Oh.
Hang on. Come out again.
Yeah,
Ooh, yeah.
There.
That's it.
nicely done.
Well done.
It's a little more than we bargained for, I think. But
That's all right. That still works. It's still helpful. Do we have a specimen number on that?
007.
007. Great. I'd like to get a Niskin. I'd like to fire a Niskin here, if we can.
we can.
So far, we've only fired one Niskin, right?
Yes, that's correct. Just
Okay.
one. And that's in the bottle 7.
Okay.
Which one did we fire?
7.
Going for 8.
Alrighty.
We have a number of scientific partners who request pieces of
You
our
got a
biological
view of it, do ya?
samples.
You're all over it.
And then the remaining go
Say
to
what?
Harvard University's Museum of Comparative Zoology, where they're stored.
I forgot how to just talk to you.
Can you see it? Can you?
Okay.
Not seen it go. But you should have activated it by now. Oh, you got it.
That was
Got it.
missing number
Got it.
2 for this dive. It's
Thank
number
you.
8.
That was way more difficult than it should have been.
That was super easy. I can't remember how to adjust that wrist thing, so using it in position mode.
Okay, so where are we in position along this slope to what we want to be looking at, Guy?
Did we get to 2C? I don't think we did yet.
Okay, so could we head down slope towards 2C?
Heading down slope towards 2C.
So we're interested in collecting bamboo corals for a few different reasons. One is that they're really difficult to tell apart, and we have lots of questions about the taxonomy of bamboo corals. And so by collecting them, we can look at them in the lab. A lot of corals, you can't tell them apart until you look at them under a microscope. So they'll help us tell species apart. Also, we're working with scientists who are interested in looking at the skeletons of bamboo corals to look at how you can look at their skeleton like tree rings and tell how old they are, and also look at, they incorporate into their skeletons and into their proteinaceous nodes the environments that they've been experiencing, both what the food that they've been eating and the water that they've been surrounded by. So they're a really interesting species to study, really interesting taxonomic group to study.
And it's so cool, just from one specimen you can learn so much about the environment too.
They can live to be about 400 years.
2C is to my south, is it? Due south?
Yeah. Actually, Dan, you want to go about 205.
205, Raja.
Can we zoom in on that coral?
Sure.
Ready, Dan? Okay.
This is another bamboo coral.
It looks very similar.
Mm-hmm. Yeah, it's pretty difficult to see the base there and the nodes, but we got some good looks at the polyps.
Yeah, that's all the zoom I got.
Okay, well let's zoom out and keep moving down slope then.
Let's pulling, coming wide,
Roger, thank you.
full wide.
Thank you. Hopefully that's the last of the mud coming off the porch.
I think he collected a little bit more on that last stop.
No, it was somewhat harder than the
Oh, okay.
stuff we've been looking at.
Maybe not.
So I actually need to, if I'm going to stay on the cliff, go look west, yeah?
Yeah, southwest at this point. It's
Gonna
about a 225.
disrupt the viz here for a minute while we change heading, look the other way.
Now you're heading north.
That's not going to work. Sorry, I'm going to have to do it again. Trying to keep the cliff in view and back down. So.
Just going for a full tether wrap too?
I hope not. This one's backwards, isn't it? It's, if I come left, it takes tether wrap out.
Left is negative tether wraps.
So that's the way I want to go.
Negative tether wraps.
Yeah.
Roger.
You're at 0.6.
I think it kind of flattens out and we get back into the soft stuff down here.
It's right behind you now.
How far are we from 2C?
13 meters.
13 meters from 2C, so it's right behind me.
That's pretty good.
Yeah.
Close enough?
Yeah, let me
you wanna
see
be
what
playing
we
on it.
want to do next.
2D is 425 meters away.
So as we continue to make our way to our next point, just want to tell new viewers that we are still in Box Canyon. We're in the Cordell Bank National Marine Sanctuary.
Wow.
This is located about 35 miles from Point Reyes off of California's coast. And over the next week, we'll be in Cordell Bank and Greater Farallones National Marine Sanctuaries. We're looking at deep canyons, excited for lots of corals, sponges, and all of their associates.
So while we're waiting, there's a bit of a cliff 20 meters to this heading. Can I make it?
Coming up to a cliff here. I wanted to just add that one of the things.
Oh yeah, there it is. Wow.
Since we've gotten into these deep water canyons in the last 2 years, it's kind of really expanded the
following
habitats
the
and species.
Mesotech nose there. So that was about 20 meters to the southeast.
What I was saying is getting into these deep canyons has really expanded our species list for the sanctuary quite a bit.
It sure has. This is an interesting feature, almost like a little cave there. So can we get a zoom on that bamboo coral that's in the bottom of the screen, just below that Pacific flat-nosed fish?
Can do.
Good. Oh, I see it now. Oh, it's coming straight at us.
Danny, can I ask you a question?
Sure.
What do we get in the Niskin bottom? We get a water
Wayne,
sample,
you can
and what
zoom
are we
in a bit
sampling
there.
for?
Alrighty. Zoom in.
We seem to be doing that near species that we're collecting. So we just collected a bamboo coral,
dead,
we
yep.
did a Niskin.
There's no.
This is something dead something. I don't know what it is. So we don't need to spend a lot of time with it. You can zoom out, and it looks like a dead bamboo coral potentially. When we take a Niskin sample, we are getting two things. We're getting a water sample for eDNA, so animals in the ocean slough off cells from their bodies. It goes into into the water, and you can sample that water for DNA for animals that are around there. Even if you don't see them or sample the organisms, you can get their DNA and understand what's in the area. We're also looking at water samples for carbonate chemistry, so we can look at the chemistry of the ocean and the what's called the aragonite saturation,
There, Wayne.
which is how
He's really close.
a level of chemistry to indicate whether the conditions are favorable to organisms who use the calcite in the carbonate chemistry in the water to build their skeletons or not, such as corals. And that can tell us about the if those conditions are favorable for that kind of growth of their skeletons.
Thank you.
So in general here, the plan is that we want to start moving to point 2D. So that is moving around this 355 meters away, moving around this promontory feature.
So you want to stay along this cliff face as long as possible before heading south and downhill?
I think we should probably try and follow along this contour, Guy. What do you think?
All of these cliffs are going to be finite, so if you're trying to follow contour, you're going to get back in the mud at some point. But yeah, since we're on rock right now, yes, follow this contour until it peters out.
Okay.
Roger. And 2D looks to be about 75 meters deeper, so we may just have to blue water it down there.
Okay. Another bamboo coral, pretty similar to the ones that we've been looking at, and one that we collected.
Nav, at some point I'm going to move site 2E. You let me know when you want those numbers.
I can do that right now. Go.
Okay.
Guy, I'm ready whenever you are.
All right, coming wide.
Thank you.
It's kind of like snorkeling.
Okay, Nav.
In a different
do you want
way.
a new target or you want to update 2E? I can do either.
I'm just going to change 2E because we're not going to go there.
Roger.
Negative 123.623.
That's it? Okay. Negative 123.623.
38.046.
38.046.
Okay, so we want to move along this wall, if we could be a little bit closer to it, or along this feature, and start moving laterally along this rocky face. That'd be great.
Copy that.
Does that look right?
Where'd the rock
We
go?
came back to the east of the little hog's back on this hill in front of us.
Yeah, I lost it. It's up above me.
You want to be over here?
As Herc flies over the seafloor, we're seeing lots of brittle stars, which are very common to this area. When we see species, new species to this region, is that something that is, I guess, a sign of something good, sign of something bad, or just a change?
A lot of the new species that we are identifying for our sanctuary are a sign that it's very underexplored. So when we identify new organisms, it's likely that they've been here for quite some time and nobody has ever looked here to see them.
When we see trash, though, that's not good. And we have seen some trash before. I remember our first dive in 2017. The first thing we saw when we landed was a plastic water bottle.
171.
Oh, bummer.
It's kind of a reminder that we have the reach, even though we can't get there personally. Our reach goes very far.
Yeah, I think we discovered a plastic 5-gallon bucket
Yes.
on earlier today.
I saw it in an earlier watch. But also a reminder, plastics were just invented in the 1950s, and so we've innovated in so many other realms, and there's real potential for some solutions around us innovating with biological materials and limiting our need for single-use plastics.
Looks like some zooplankton swimming around
get
here.
It going and then, yeah.
Or is that just the ROV moving? I guess the ROV's moving.
Yeah, the DP PID control seems to do it a little bit better. Trecaline will sit there and try and get you online before going, which is weird.
On one of our surveys last year on a different vessel with a different ROV, we collected a coral specimen from Cordell Bank itself, the bank that's in a shallower region than where we are right now. And Gary Williams, who's on board with us, described that as a new species. It's a little yellow coral, and he named it Chromoplexora cordelbankensis. So there is a coral named after Cordell Bank.
Oh. Apropos.
Okay, so we want to explore this hard substrate, looking for some different coral species. And as we move along this wall, we can stop and take a look. We're looking for bamboo corals, black corals, some sponges.
I'm going to come up the wall just a bit while waiting for the vessel to catch up.
Okay.
You all right with that?
Yeah.
See, I'm tugging on Argus there, so. Gotta be something hanging out here worth looking at.
Someone tuning in asks the question, if we come across an organism that's particularly rare, how do you make that decision on whether or not to sample it?
Yeah, so we are generally looking at, as we're moving along, looking at how many times we're seeing some of the different corals, and we generally don't collect something that we haven't seen before and it's the only one around. So we'll likely want to see like 3 or 5 specimens before we collect. It sometimes is appropriate to collect something that we've only seen once. Sometimes that we know it's we're not going to come across it very often. Let's take a look at those bamboos if you can get over there.
Copy that.
We put a lot of thought into what we want to collect, what our targets and objectives are for the cruise, and we think about what we actually need, what we already have sampled before and have well identified and well-known species. So we try and be really thoughtful about that because we are collecting in a national marine sanctuary. We have to get permits from ourselves to do that. And we definitely don't want to be depleting these species. However, these surveys are really covering us just a snapshot of the bottom of the ocean, and there's a lot more habitat out there. So it's likely there's a lot more organisms than the ones that we see on these surveys.
Hey Guy, are these concretions sticking out of the wall?
Yep, they are.
All right, zooming in.
That is really cool. You can finally see that.
Zooming in.
Guy, can you expand on what we're seeing?
All right.
Up above the bamboo coral.
I can come wide.
Well, without a hand specimen, I can't for sure. It's either a limestone that's precipitated within a more porous limestone, so it's just more indurated and dense than the surrounding limestone. So it actually forms by fluid flow through the limestone around some kind of small seed clast, like a shell. But it could also be a mud inclusion. So there's several ways you can get these features. And I don't know which one this is.
That's it. Coming wide. Alright.
Hey, Roger. I've pulled Argus around again to get that shot, so we'll wait for the boat.
Oh, we're gonna be here for what?
No, Antonelle's coming back around, but as you can see, our leash is tight there. We only have 30 meters.
It's interesting what's under that ledge to the left. Those little things.
Looks like we've got some worms there, some worm tubes. Some of them are pulled in, but you can see some of the feather dusters there.
Oh yeah.
I'm touching there, Wayne, if you want to zoom in.
Okay, I can give it a try.
I'll have to move the big camera there.
Yeah, I was. Oh, I see. Yeah, we scared them off. That's all right. That's all right.
Might be a good Halloween costume.
And there's a picnigonid sea spider.
All right, try again.
Okay. Let's see. Yeah, I think that it's just an anemone, but it looked like a coral for a second when I just caught a glimpse of it when it was in the shadow. It's just an anemone. So I don't think there's.
I hit too hard and they
No,
retracted.
no, I don't think there's much to see. Those are just the sea pans.
Oh, the tube worms there.
Yeah.
I bumped and they.
Yeah. Oh, now I see the spider. Boy, spindly. What? Wow. There's a star hitchhiking a ride on a spider. You don't see that every day. Okay, coming wide. Nice. A little more.
163 you're moving? 165.
Just reading up a little bit on the sea spiders, the picnigonid feeds on soft-bodied invertebrates, in particular cnidarians, sucking at them with their proboscis.
Anybody
And
home?
larval picnigonids often live as parasites within cnidarian tissues.
You want a partial
Very
zoom?
cool.
Sure.
Whoa, photobomb.
Can you let me know when we're ready to move again?
We are moving.
We're moving so we're able to move the ROV?
Just now getting a little bit of slack there to move on.
Okay.
Oh,
Let me know when you're
Who's
ready to
that
start
guy?
moving along that wall.
that guy's got a little bioluminescence going on. That little.
Oh, it's reflecting the light from the ROV.
Maybe.
Always.
Takes a minute for Argus to get moving as well, so 2 clicks away from the ship. Moving along the wall.
Great. Oh, looks like that might be a Welk tower off on that ledge concretions there. So there's a Welk. So these Welk lay eggs in these towers.
Oh, the chimney white
Yes.
looking guy? That guy.
Which is a snail, right? So the snail, does it
Yes.
live in there?
Those are the eggs. Makes an egg tower.
Oh, and that's the whelk.
Yes.
Beautiful.
Assuming.
Oh, let me get in on it.
That is cool.
It's an impressive little structure he's got there.
Right?
We saw an area off Morro Bay that was just packed with those.
Oh, yeah?
It was amazing. Thousands of them.
Wow. It's got a...
Off we go.
Oh.
Popular hotel there.
Yeah.
This is really interesting habitat with all these little crevices.
And these are serpent stars.
These are the Brycingids.
Brycingids.
I have a hard
Oh,
time
the
saying it.
serpent is a little bit bumpier in the middle, right?
And there's a soft coral, the Heteropolypus, and a predatory tunicate. We've been seeing quite a bit of those.
Okay, coming wide. Full wide.
You can zoom back in just a bit. That was a good zoom level there.
Okay.
So the lasers are 1/3
Yeah.
down the screen. Yeah, right there. Perfect.
All right.
There's actually a lot of Halloween costume inspiration right here. I'm just deciding, should I do predatory tunicate or one of those tube worms?
That looks great. Let's see if there's anything in that little cave that's up above.
Is that a glass sponge? Or what's the... just to the right? I don't know. Oh.
Contact. Cliff contact.
Full wide.
I got a little close there, sorry. It's evidenced by the debris raining down into the picture. If you're touching, you're too close.
That's a good general rule.
Sorry, Danny, I was gonna get the in the cave shot, but I dusted it. So.
That's all right. It didn't look like there was too much in there.
Ooh.
Let's take a look at that bamboo coral.
Okay.
Go ahead, Wayne.
Oh, and there's another one.
That's good.
Actually, we might be able to get a better look at the base to the one on the right. This is going to be...
Hold the zoom there for a minute. I'll,
okay, hold it.
Try and put it in the light.
All right.
For a shot. Okay.
All right. All right, let's
Zoomy zoom.
push in.
Good.
Yeah. It's wearing a skirt.
Yeah. Some sponges around the base of it. It's a little hard to tell.
Yeah. Right around where we need to see.
Yeah. So let's, if we could turn to just the one out of screen to the right. I might get a
Copy,
good look at
coming in.
that.
Yeah.
I have to move a little slower here, otherwise they dust the wall.
Right.
Yeah.
They want the visibility to.
If we could get a profile of it, and
Can do.
I don't know how.
Yeah, come right.
Oh wait,
this
we got
gets
the
against
base right
wall.
there. We got the base right there. We can see it. That's definitely what we need to see. I just need to get it in focus.
We like to look at the base and also the branching and where that branching occurs.
Yeah, that's a good shot.
And why is the base and the branching important to capture?
They can be distinctive, and especially the branching, whether they branch at the node or internode, has traditionally been thought of as a characteristic feature, although there's some discussion about whether that is the most definitive feature to look at or not. So we want to look at some different morphologies, and we've already made 2 bamboo coral collections. One of them was similar to this one, so we can look at that as well as get some close-ups of these images that can help us start to distinguish them.
Now, those fine hairs that you see, are those part of the
Just a little focus, Wayne.
the part of the coral, or
Thank you.
Yeah,
is
those
it-
are likely some defensive
okay.
features that they use to is kind of sweeper tentacles to keep
Contact.
other organisms away.
Okay, I'm touching the cliff with the bumper there. You can
Gotcha.
zoom out.
Okay, coming wide.
It's as close as I can get.
That was okay. Full wide.
Let's have a peek here. Let's, judging by the sonar, it goes on for a ways. So.
Great, we'll just keep following this along.
Copy that.
Do we have plans, Danny, to collect any geological samples, push
Ooh.
cores or rocks along this transect?
Yeah, go ahead, Wayne.
All right, pushing in.
We don't have any plans unless we see something that Guy wants.
That is.
Guy, could those denser pieces of limestone be rocks from land?
Well, yeah, they could have formed it on limestone that was above sea level, yeah.
Busted her.
Any limestone that had water flowing through it. I'm assuming that they're concretions in a limestone, but I don't actually know for sure if they are.
Moving
Coming
on.
wide, coming wide.
They look tumble worn.
No, I don't think they were in a river at any time. These are things that formed after the rock had already formed.
It looks similar to what you see poking out of Agate Beach in Oregon. Then they have, those ones you crack and have a fossil inside. Ooh.
It does seem to be in a layer.
Would you like to see the bamboo down there?
Oh, little jelly.
Guy, can you speak to how these deep canyons were formed and how old we suspect them to be? as rock formations.
Can you decrease the range on the
Decrease.
Herc to 5 meters?
Well, the canyons are
Thank you.
just the result of uplift of the continent, and then slumping initially would have been kind of random, but eventually those sorts of slumping coalesce into sort of evenly spaced canyon features, and we see it all up and down the coast. I do not know how old they are, though. I haven't studied that.
One of the benefits of being national.
Oh, let's take a look at
Ooh.
that primnoid.
Okay.
I'll come around and give you the
All right.
broadside.
I'll leave you wide.
You can zoom in just a bit there if you
Okay.
want.
Coming
It's
in.
a really neat shape.
Yes.
Yeah, perfect right there, Wayne. Give me a little wiggle room.
It's a primnoid coral, potentially Peristanella.
You think it's growing that way to take advantage of the current?
It looks like it is, doesn't it? And it kind of has a cup shape to it, which is fairly common for that species. Also common to see brittle stars on it like that. I always think of them as being decorated with the brittle stars.
Let's see. Get a little closer there.
I want to call those spoon worms tongue worms. They kind of look like little tongues.
Yeah. Okay.
Thank you.
This.
Really
Excuse
cool.
me.
We'll come
Full
around
wide.
and look at the back side. See if I can park the other side.
Okay.
Beautiful.
Yeah, it's
So intricate.
that's
full zoom there.
yeah. That's full zoom.
Just gonna try and get in the base, but just
Yeah.
not quite right.
Well, that's a great shot. We know this one fairly well. We don't need to collect it. So it's, we can move on.
Okay, coming
Moving
wide.
on.
Well, that wasn't very much. I was right on top of it.
Yeah, I'm trying to use the bull bars up there to
Oh, I see.
park on the cliff.
Okay.
Kills the viz a little, but it gives you the stable
This,
shots.
yeah, it really.
Straight off.
I wonder how old that coral is,
Not quite
knowing
hard
how
enough
slow they
cliff
grow.
to-
That's a pretty
So
intricate,
it took me-
large specimen.
Yeah, it looked pretty robust.
What I wanted to share earlier was one of the benefits of being a National Marine Sanctuary is there's a lot more effort to understand these areas. So mapping has been a priority in National Marine Sanctuaries, and we would not be here if it wasn't for the mapping that's been done to better determine where to go to research. With less than 5% of our seafloor mapped in the entire world's ocean, it's nice to have these special places that are target areas to map and better understand, to learn more about the ocean habitats.
Yeah, we have some good seafloor maps of these areas thanks to NOAA's Office of Ocean, the Okeanos Explorer mapped this area. And then the Nautilus remapped some of those areas to look at a little more fine scale. So we actually have good seafloor maps of this area. Well, it's interesting. We're not seeing a ton of corals here, but the corals that we are seeing are primarily bamboo corals. We did just see a primnoid, but here's another bamboo coral up ahead.
I'm going to zoom in.
Yes.
Okay.
Okay.
Which one do you want to look at there?
The white one. The yellow one is a stalked crinoid.
Pushing in.
Copy.
Okay, getting that in focus.
Another one of those purple dudes.
That one looks a bit thinner than some of the others. I don't know. Maybe it's just further away.
Okay, we can zoom out.
Okay, coming
Keep
wide.
going.
For what?
Okay.
And although this area has been fairly well mapped recently, more than 80% of our world's oceans are unexplored and unmapped, and EV Nautilus is participating in the Seabed 2030 effort to map the gap, so essentially help map the world's seafloors.
Okay, pushing in.
We've got, that's another one of those sponges with some crinoids on it.
Okay, nice.
Going for the Z bias to get those just smooth kind of shots. She never touches that lever. She gets it perfectly.
That's the way you do it.
What's that?
It's a way to do it.
That's the way. Yeah, especially doing this now. We're not going up and down, so.
Hi, Morgan Patton from EAC. Thanks for tuning in with your daughter. We're glad you guys are watching.
Hi, Morgan.
Yeah, so what we're doing, we're dialing the verts in to hold the vehicle level, so you're not trying to, and then to go up, when you pull back, you're just doing less vertical. And to go down, you give it a hair more. Then when you let go of it, it tends to hold itself there long enough for the shot, other than trying to chase it. So by putting the trim in, or the Z bias as it's called, we can get a smoother shot. Than the machine flies better than the human. So. Yeah, then I'm just doing the heading and.
One of our viewers was curious why we are seeing so few fish in this area.
Yeah, we haven't seen a ton of fish. In general, the density of fish in the deep sea is less than if you're thinking of more nearshore species, like the coastal environments. That there are fewer fish species and fewer and far between. We have been seeing Pacific flatnose, eelpouts, and grenadiers. We saw skate earlier. So we've been seeing a few, but not a ton. There's also, because we're on this really high slope cliff habitat, sometimes we might be more likely to see fish rest. A lot of these fish will rest on the bottom, so we might be more likely to see them if we're on a flatter habitat.
Or near a community of sponges and corals, and we seem to be seeing a lot of isolated corals, isolated sponges, as opposed to communities which really form a structure for fish to hang out around.
That's what I don't like about the Z bias. I give it too much and it keeps
Kill.
going.
One of our viewers was also just curious about our watches and how we prepare for them. So our watches are structured on 4-hour intervals, And we switch therefore every 4 hours, and a new watch team comes in.
Yeah, and there's three sets of there's 3 teams that take turns doing their 4-hour shifts. So if you work from, for instance, we have a midnight to 4.
So there's a few things to look at here. Let's take a look at that sponge that's just ahead of you. We got kind of the bigger version of this before, the Calophocus, with some pieces of it, and this could potentially be a smaller version of that.
This guy here under the lasers?
Nope, to the left.
Sorry.
Don't.
Let me come wide.
This one.
Yeah, that's the one. Coming in.
That's a good shot of that. We don't need to collect this one, but we'll get some pictures.
Okay, it can come in a little more.
Yeah, we back up a little.
Get it in focus. Oh yeah, there. Okay, our distance is increasing now from it.
Too far? Oh, it is.
That's
Sorry,
okay. That's all right. That's all right. I think we got what we needed.
I'm chasing the zoom.
Yeah. Okay.
So those rocks projecting now look like there probably are a layer that's been stretched to the point where it's no longer continuous. A lot of these rocks are really extremely deformed. It's a real slow process, but they can be really mangled.
Yeah.
Okay, we got that. Thank you.
Okay, coming wide.
What else do you want to see here?
That's it for here.
Okay.
All that dirt on the porch.
Who did that?
Oh, some farmer pilot.
Wayne, do you mind continuing to fill listeners in on how our watches are organized?
So yeah, there's 3 teams of pilots and scientists that work as a group. I guess if I count them real quick, 1, 2, 3, 4, 5. 6. There's 8. There's 8 of us. So there's 3 groups of 8 scientists.
Getting a little stretched out.
Which is 20.
Can we take a look at that bamboo coral that's just to the left of the lasers, please?
Can do.
Yeah, so that'll be 24 scientists in 3 groups of 8. And if you work the midnight to 4, you also work the noon to 4 p.m. shift.
Yep, so this allows us to work around the clock and to achieve the objectives of each dive. So this current dive in Box Canyon is expected to be about 20 hours. We started at 9 a.m.-ish Pacific time.
Yeah, you're north of the ship now.
Full wide.
Oh, I can stop.
Yeah.
You're not on SPL. They're on SPL.